Welcome back to our biochemistry battlefield, and today we are diving headfirst into a topic that's often overlooked but is as fundamental as a good chai: carbohydrate chemistry. Forget just sugar. These are the master architects, the energy currency, and even the identity markers of our cells. Picture this: a young patient, let's call him Rohan, a bright eyed 6-year-old from a small village in Rajasthan, comes to the OPD with persistent diarrhea, bloating, and abdominal pain after drinking milk. His parents are worried. They tell you he's always been sensitive to doodh. You, my friend, immediately think carbohydrate malabsorption, specifically lactose intolerance. So, what are these magnificent molecules? At their simplest, carbohydrates are polyhydroxyaldehydes or ketones, or substances that yield these upon hydrolysis. Their empirical formula is CH2O. This gave them the historical misnomer hydrates of carbon, as if water molecules were simply plastered onto carbon atoms. But no, it's far more elegant. Think of them as the versatile Lego bricks of biology. From the simplest monosaccharide, a single brick, to the most complex polysaccharides from the rice in our dal chawal, they are our primary energy source. Let's begin our journey starting with the simplest units. The first one is monosaccharides. These are the fundamental building blocks. Monosaccharides are simple sugars, the unhydrolyzable units. They are classified by two main criteria. First, by the number of carbon atoms, trioses, which has 3 carbon atoms like glyceraldehyde, tetroses, which has 4 carbons, pentoses 5 carbons like ribose in DNA or RNA, hexoses 6 carbons like glucose, fructose, and galactose. The second is by their functional group. So aldoses have an aldehyde group, example glucose and galactose, or ketoses with a ketone group, example fructose. Remember, a simple way to recall this: aldoses have an aldehyde, ketoses have a ketone, right? It's easy. Now the fascinating bit: chirality. Most carbons in a monosaccharide, except the anomeric carbon and the terminal carbons, are chiral centers. This means they have 4 different groups attached, leading to stereoisomers. In biology, we predominantly encounter D isomers, where the hydroxyl group on the penultimate, which is the second to last carbon, is on the right in a Fischer projection. This D or L designation is absolutely critical. Enzymes are like highly specialized tailors. They only recognize specific shapes and orientations. Imagine trying to wear a left-handed glove on your right hand. It just doesn't fit, right? This stereospecificity is fundamental to biochemical reactions. For D sugars, if the anomeric hydroxyl group is down in the Haworth projection, it's alpha. If it's up, it's beta. Think of it like a namaste gesture. Alpha bows down, beta stands tall. This seemingly small difference is huge for how enzymes recognize and interact with these sugars, and how they link together to form larger structures, influencing everything from digestibility to structural rigidity. Next, we talk about the concept of reducing sugars. Any monosaccharide with a free anomeric carbon that can open up to its aldehyde or ketone form is a reducing sugar. This means it can donate electrons and reduce other compounds, which is a property we exploit in clinical labs for tests like Benedict's or Felling's, which detect glucose in urine. A simple rule: if it has a free hemiacetal or hemiketal group, it's a reducing sugar. Next, we come to monosaccharide derivatives. These are monosaccharides with chemical modifications, making them even more versatile. So the first one is amino sugars, like N-acetyl glucosamine and N-acetyl galactosamine. Here, a hydroxyl group is replaced by an amino group, often acetylated. They are the building blocks of chitin. Chitin is present in crab shells or the cell walls of fungi, which is important in fungal infections you'll see in rural practice. And bacterial peptidoglycan, the target of many antibiotics like penicillin, which is a true clinical pearl here right now. So the first one was amino sugars. The next one is sugar acids. These are formed by oxidation like glucouronic acid. They are crucial for detoxifying xenobiotics in the liver. They are also useful in conjugating bilirubin and forming components of connective tissue. The third one is sugar alcohols. These are formed by reduction, like sorbitol. These are used as sweeteners, but in diabetics, accumulation can lead to osmotic damage, contributing to implications like retinopathy and neuropathy. Now that we have discussed about monosaccharides, we move to disaccharides. Okay, these are the dynamic duos: 2 bricks, 1 strong bond. So let's take two of these monosaccharide bricks and join them together. When two monosaccharides link via glycosidic bond formed through a dehydration reaction, which is removal of water molecule, they form a disaccharide. This bond is catalyzed by specific glycosyl transferase enzymes, which are like the skilled masons of our cellular construction site. The beauty and complexity lies in the specificity of this bond, which anomer, alpha or beta, is involved, and which carbons on each sugar link up. Example 1 to 4, or 1 to 6, or 1 to 2. This seemingly small detail profoundly impacts the disaccharide's properties and how our body can digest it. So, now we come to the examples of disaccharides. The first one is sucrose, or table sugar. This is the quintessential chini or sugar which you put in your chai. It's glucose. Alpha 1 2 fructose. What's unique here? Both anomeric carbons are involved in the glycosidic bond. This makes sucrose a non-reducing sugar because neither sugar can open its ring to expose a free aldehyde or ketone. It's the primary transport sugar in plants, moving energy from leaves to other parts. Next one is lactose, or milk sugar. So this is crucial for neonates. It's galactose beta 1,4 glucose. This is a reducing sugar. Because the anomeric carbon of the glucose residue is free. Hydrolysis by the enzyme lactase in our small intestine is essential for digestion. A deficiency in lactase, common in many adult Indians, leads to lactose intolerance. Our Rohan from Rajasthan's problem. Undigested lactose ferments in the colon, causing gas, bloating, and diarrhea. Next one is maltose, or malt sugar. This is glucose alpha 1,4 glucose. It is again a reducing sugar. It's often an intermediate product when starch is broken down during digestion. Think of it as a stepping stone from complex carbs to simple glucose. Next is isomaltose. So glucose alpha 1,6 glucose. It is also a reducing sugar formed at the branch points of starch and glycogen. Next is cellobiose. This is glucose beta 1,4 glucose, formed from cellulose breakdown, but we lack the enzyme to digest it. This beta linkage is why we can't digest cellulose. Finally, there's lactulose, a synthetic disaccharide. It is galactose beta 1,4 fructose. It is not found naturally. It's a non-absorbable sugar used clinically as a laxative and to treat hepatic encephalopathy in liver failure patients. It works by acidifying the gut, trapping ammonia and reducing its absorption. A clever trick using carbohydrate chemistry. Next, we come to oligosaccharides. So these are the cellular ID cards. Short chains, big rolls. Moving up in complexity, oligosaccharides are short chains, typically 3 to 10 monosaccharide units. While not as abundant as monosaccharides or polysaccharides, their role is disproportionately vital, especially in cell recognition and communication. Imagine them as the ID cards or signaling flags on the surface of our cells. They are most often found covalently attached to proteins, forming glycoproteins. The precise sequence, branching pattern, and type of monosaccharide in these oligosaccharides create an incredibly diverse and specific molecular signature. This molecular barcode allows cells to 1. Cell-to-cell recognition, 2. Cell adhesion, 3. Signaling, 4. Blood group antigens, and 5. Pathogen recognition. So all of these are possible only because of these oligosaccharides. Next, we come to polysaccharides. These are the grand structures and they provide storage and support. Now let's assemble hundreds, even thousands of monosaccharide units into long chains or branched structures. These are our polysaccharides. They are the grand structures serving either as efficient energy storage depots or robust structural components. We classify them as homopolysaccharides, which are made of a single type of monosaccharide, or heteropolysaccharides, made of multiple types, often repeating disaccharide units. So in polysaccharides, the first type of polysaccharides are the ones which store energy. Now these are designed for compact, efficient storage and rapid mobilization of glucose. The first one is starch, which is present in plants. This is the primary energy reserve in them, and it is the staple of our rotis, chawal, and aloo. It exists in two forms, amylose and amylopectin. Amylose is a linear polymer linked by alpha 1,4 glycosidic bonds. It forms a helical structure like a coiled spring. Amylopectin, on the other hand, is a highly branched polymer. The main chain has alpha 1,4 linkages, but every 24 to 30 glucose units, an alpha 1,6 linkage creates a branch point. This branching is crucial because it provides multiple ends for enzymes to attack, allowing for rapid glucose release. Next, there are dextrins. These are smaller glucose polymers formed during the partial hydrolysis of starch by enzymes or acids. Next, there are dextrans. These are synthesized by bacteria. These are highly branched glucose polymers with alpha 1,6, alpha 1,4, and alpha 1,3. Technically, they are used as plasma expanders in emergencies, and they are a lifesaver when blood volume needs to be quickly restored, for example, after trauma or severe burns. So, after starch, which is the main primary storage energy in plants, we come to glycogen, which is the primary storage energy in animals and fungi. So, in human beings, stored primarily in the liver and muscle. It's structurally very similar to amylopectin, but far more highly branched, alpha 1,6 linkages every 8 to 12 glucose units. This extensive branching is a master stroke of evolution. It allows for rapid synthesis, which is glycogenesis, and breakdown, which is glycogenolysis, ensuring quick access to glucose for metabolic demands. Liver glycogen maintains blood glucose levels for the brain and red blood cells, which are basically our VIP organs. While muscle glycogen provides immediate energy for contracting muscles during that intense gym session or a sudden sprint to catch the local. Next, we have structural polysaccharides. These provide strength, rigidity, and protection. So the first one here is cellulose, which is present in plants. It is the most abundant organic molecule on Earth, forming the primary component of plant cell walls. It's a linear homopolymer of glucose units linked by beta 1,4 glycosidic bonds. These beta linkages are a game changer. It forces adjacent glucose units to flip 180 degrees relative to each other, allowing extensive intermolecular hydrogen bonding between parallel cellulose chains. This creates strong, insoluble microfibrils that provide immense tensile strength. Think of the sturdy trunk of a banyan tree. Humans and most mammals lack the enzyme beta-glucosidase, which is used to hydrolyze these beta 1,4 bonds. So while we eat a lot of plant material, cellulose acts as a dietary fiber, providing bulk for peristalsis. But next is chitin. This is present in fungi and arthropods, the main component of fungal cell walls and the exoskeletons of the insects and crustaceans like the crabs and prawns you might enjoy. It's a linear homopolymer of N-acetyl glucosamine units linked by beta 1,4 glycosidic bonds. The last one we are going to talk about is peptidoglycan, which is in bacteria. It is a unique heteropolymer that forms the rigid layer of bacterial cell walls. It consists of alternating N-acetyl glucosamine and N-acetyl muramine linked by beta 1,4 bonds, with peptide crosslinks providing additional strength. This structure is vital for bacterial survival and is the classic target for penicillin and other beta-lactam. And next we come to heteropolysaccharides. Now in heteropolysaccharides, glycosaminoglycans, or in short GAGs, these are the most important ones. These are often called mucopolysaccharides and are composed of repeating disaccharide units, usually containing a sugar acid like D-glucouronic acid or L-ioduronic acid, and an amino sugar like N-acetylglucosamine or N-acetylgalactosamine, often with sulfate groups. They are highly negatively charged due to the carboxyl and sulfate groups. These groups attract water and form hydrated gels. They are the cement of a connective tissue, providing lubrication, shock absorption, and structural support. Finally, we come to clinical connections. Let's look at some direct applications you'll encounter in your practices. So the first one is the most common disorder, it's diabetes mellitus. It's fundamentally a disorder of glucose metabolism. And whether it's type 1 or type 2, the common thread is dysregulation of blood glucose levels. Next is lactose intolerance. Now, as with Rohan, who we talked about in the beginning, this is extremely common in adults across India due to declining lactose activity post-weaning. Patients presenting with abdominal cramps, bloating, and diarrhea after consuming milk products. Diagnosis often involves a hydrogen breath test. Well, lactase supplements or simply avoiding lactose. It's a direct consequence of a specific disaccharide not being high. Next is glycogen storage diseases or GSDs. They are genetic disorders where specific enzymes involved in glycogen synthesis or breakdown are deficient. Example is von Guericke's disease. It is a deficiency of glucose 6-phosphatase enzyme and it leads to severe hypoglycemia and a host of other problems. Then we have Mucopolysaccharidosis or MPS. These are a group of inherited metabolic disorders caused by deficiency of lysosomal enzymes required for degradation of glycosaminoglycans. So undegraded glycosaminoglycans accumulate in various tissues leading to progressive cellular and organ damage. Example, Hurler's syndrome is MPS type 1, and Hunter syndrome is MPS type 2. It involves accumulation of dermatan sulfate and heparan sulfate, leading to skeletal deformities, coarse facial features, early diagnosis, and enzyme replacement therapy, even though expensive, can significantly improve outcomes. Then the bacterial cell wall targeting, we talked about peptidoglycan. So beta-lactam antibiotics like penicillin work by inhibiting the synthesis of this bacterial cell wall component. It's a very important antibiotic. These examples underscore that carbohydrate chemistry isn't abstract. It's the very foundation of diagnostic tests, therapeutic interventions, and patient management in every ward you'll step. Alright, now let's make sure these concepts stick, okay? So the first one, I want to give you a couple of mnemonics to help you remember these concepts. So the first one is mnemonic for monosaccharide classification. 3 tigers play happily. Trioses, tetroses, pentoses, hexoses. Okay, the next one is mnemonic for key disaccharides and their sugars. My lovely sweets. Maltose, lactose, sucrose. Now, maltose is basically glucose plus glucose, like 2 goli soda bottles joined together. Lactose is galactose plus glucose, like milk from a cow. Galactose is for gavaiya. And sucrose is glucose plus fructose. Table sugar, the GF of sugars, but non-reducing. Next is alpha versus beta linkages, the Shadi analogy. So alpha linkages are like the traditional easily broken bonds in Shadi food, like starch, glycogen, easily digested by our enzymes. Think of the mithai disappearing quickly. Beta linkages are like the strong unbreakable bonds of a truly committed these are cellulose and chitin. Very difficult for enzymes to break. That's why fiber is so tough. Okay, the next one is clinical case checkpoint. Remember Rohan, our lactose intolerant patient? What enzyme was deficient? Exactly, lactase. What kind of carbohydrate was affected? Yeah, disaccharide. See, you're already thinking like a clinician. Now, if you found a new plant that was incredibly sweet, but your patient couldn't digest it, what kind of glycosidic bond would you support? Right, think beta linkage like cellulose. Finally, we come to a high yield summary for you all. First one is carbohydrates are polyhydroxy aldehydes or ketones. Their structure dictates function from simple sugars to complex polymers. The next one is chirality and anomerisms are key. D isomers, alpha, beta anomers, and specific glycosidic linkages are recognized by enzymes and biological activity. Monosaccharides are fuel, disaccharides are transport, oligosaccharides are identity, polysaccharides are storage or structure. Each class has distinct vital roles. The clinical relevance is immense, from diabetes to lactose intolerance, glycogen storage disorders, MPS, and antibiotic action. Carbohydrate chemistry underpins countless diseases. You have now got a solid foundation in the chemistry of carbohydrates. You understand their definition, classification, examples, and functions. These aren't just dry facts, they are the language of life, the building blocks of health and disease. Thank you for watching this video, and I hope to see you in the next one.